I keep underestimating plants. During my bachelor's degree, we were all very annoyed when we had to do classic botany. It was very tiresome to do all the classifications. You had so much information coming your way, just like you will in a minute, that it was quite overwhelming. We had to dissect plants, and we were just like, it's a plant, what do you want from me? But ultimately, it gave me a greater understanding and appreciation for plants that I can hopefully convey to you today. I feel like an understanding has arrived in our culture that animals can be really smart, like dolphins, or pigs are actually really smart. And there's a general appreciation for animal lives, but plants sort of go under the radar. We grow our monocultures and only eat a handful of different plants. Even tortillas are made out of wheat here. That's wheat with an A. But there's so much diversity in the plant kingdom, and with that, a great potential. And I hope I can convey to you how incredible that is. Because not only are there so many different species of plants, but they also synthesized a vast variety of molecules in so many different pathways. My professor used to say, it's because plants can't run away. What that means is, if an animal is faced with a predator or heat or cold, it can just walk away. Whereas plants have to compute the threat in molecular pathways and come up with a molecule to protect them. And it's going to be different molecules for protection against predators versus heat. Same with sponges, which are animals, but they too can't run away, which is why some of them are poisonous and are also looked at to find cures for cancer. Anyway, cannabis has come more and more into focus because, as this paper says, there are therapeutic potential for all human diseases. And because it has been legalized in more and more countries, I guess. But for thousands of years, cannabis has been cultured and selected for the psychedelic components. It's the cannabinoids like cannabidiol or CBD that have the health benefits. And that's what has been legalized in my country. It's sold as an extract that contains the medicinal molecules of CBD, but not the psychoactive THC. And now we're finally getting to the paper that we're talking about today, where Berman and al. Proved that the straw flower also contains cannabidiol-like molecules when cannabis was thought of to be the only source. But first, welcome to the club. I'm Hannah, I'm a molecular biologist, and today we're talking about cannabidiolic acid. This is a very lengthy study. They looked at so many different molecules, I couldn't possibly cover them all. So I'll focus on cannabidiolic acid, or CBGA, or CBG if it's no longer an acid. While the cannabis plant is growing, there's a lot of CBG present, but the older the plant gets, the lower the concentrations, which is why CBG is more expensive as the better known CBD, and that is probably also the reason why there's more research on CBD. But for all we know, it has the same beneficial properties. And here's where my ignorance kicked in again, because if we have a molecule and a plant that produces it, then what's the issue? Well, they only really get to this at the end of the paper, but cannabis produces these molecules only in the female flowers, which means you have to grow a lot of plant to harvest the thing that you want. And there's a season for the flowers. In fact, cannabis needs a certain light pattern that is very costly if you want to emulate it artificially, and there's often conflict in the places where it grows naturally. So it would be good to have an alternative. Moreover, these guys found that the molecules from the straw flower are modified in a way that allows our body to take them up faster. But we're getting ahead of ourselves. So they say in the introduction, Helichrysum umbracularium, the straw flower, an Asteraceae or sunflower family plant, has been used in fumigation rituals and traditional medicine by native South African tribes. More than 40 years ago, Berman and Hoffman identified CBGA in this plant, making it the only known species after cannabis from which the cannabis-like cannabinoids have been isolated and characterized. So that's interesting, but let's zoom in through those Latin words that don't mean anything if you don't know what it means, but carry a lot of information if you do. When talking about evolution and the tree of life, plants often get overlooked. Classic botany has an old school feeling, and it doesn't relate to us humans so closely. But as scientists, we're always in pursuit of knowledge, so let's take the plunge. Here you see the tree of life covering the plant kingdom. Plants, like animals, came out of the water. So it starts with moss, then ferns, and pine trees that all still need water to reproduce. Then in pink we have the monocots. Those are plants that I recognize by the parallel running ribs on their leaves. Not sure anymore if that's the official physiological distinction, but it is mostly grass and also lilies of the valley, for example. But the vast majority of all plant species are oidocoids. Compared to the monocots, the ribs on their leaves look more like a net. With not quite as much emphasis on the diversity of the oidocots is this tree. I'm just taking this from the publication Evolution of Floral Diversity, Genomics, Genes and Gamma. Here you also find the ginkgo trees, the pine trees, magnolia, lilies, and then here we have the oidocoids again. The first group that separates itself here are the buttercups, very common where I live. Then we get to the Panteptalaea. This group was distinct by genetic analysis and not classic plant physiology. It represents the last common ancestor of cannabis and straw flowers. This separation happened 113 million years ago. For reference, humans have only existed for 300,000 years, so not even 500,000 years. And within this group we find the rosids, one of the largest groups within the oidocots, comprising of approximately 70,000 species. Just for reference, there are about 60,000 mammalian species. So if you thought humans and rats are quite different, there's much more diversity here. And among these 70,000 species is cannabis. The other big group are the Asteraceae, or sunflower family. It contains about 32,000 species, one of which is the strawflower, which is the plant that they now confirmed here also contains cannabinoids. The way they did that is by mass spectrometry. They isolated all the molecules from the strawflower and ran it through the tube of the mass spec. The molecules interact with the lining of the tube and come out the other side separated according to their size and electrical charge. The computer registers them and records the spectrum, which is what we see here. The authors have annotated this peak representing the cannabinoids. Like in cannabis, there are different ones like CBD, CBG, and so on. The mass spec can also quantify the amount of CBG that is in the sample. They took samples from different parts of the plant and found that the highest concentration of CBG was in the leaves, which was 4.3%. At the bottom of this table, they list what the average amount of CBG found in the flowers of cannabis is, which is information they took from the literature. And it is a lot less, only 0.4%. And as I've already hinted at, the leaves have a lot more weight than the flowers alone, which is of course good if you want to isolate CBG and sell it. And I hope that after this detour I took you on, you can appreciate that it's really rather astonishing that the strawflower makes these cannabinoids even though they're really not related to cannabis. The fact that they're so far apart on the phylogenetic tree is also one of the reasons why the authors think that the strawflower developed this molecular pathway of building cannabinoids independently. Actually, they did more than that. They meticulously looked at the intermediate stages of the cannabinoid production with the mass spec that led them to the same conclusion. The scientific question that arises from this is, why would the development of cannabinoids happen twice in evolution? What use is it to the plant? But the more worldly question is, what good is it for humanity? Well, they also found with their mass spectrometry that the cannabinoids here have modifications, something that has never been reported for the cannabinoids from cannabis. So how did they do it? Well, they smashed the molecules that they isolated from the strawflower with positive and negative charges, so electrodes and cations. Then the samples went through the mass spectrometer, and from the pieces that came out the other side, they could deduce the additional chemical groups on the CBGA. Here you see there are two glycosylations, which means sugar has been added. And here's another potential site for glycosylation or acetylation, which is a reaction with an alcohol group. Glycosylation is a very common thing. If you have the blood type A or B, for example, your blood cells have been modified with sugars. Your body can recognize those cells as your own, but you can't donate blood to other people that don't share this blood type because their bodies will recognize it as foreign. So from a biological standpoint, it's very useful. In this case, glycosylation increases the aqueous solubility, which has been proposed as a strategy to enhance bioavailability and absorption of cannabinoids into the bloodstream, they write. Which means the cannabinoids with the sugars are more water soluble, and since our bodies are 50% made up of water, it makes it more accessible to the cells. This could mean it reaches more cells, lower dosages might be needed, and we all have different metabolisms, so it might actually be more effective than the cannabinoids from cannabis for some people. Let me know what you think in the comments, and if you have any questions. Like and subscribe if you want to see more, and check out my blog Science Shows, where I write about how scientific findings matter to me on a more personal level. The link to that is in the description. Until next time, bye!